I would like to welcome Bence György. He is assistant professor at the University of Basel and head of translation at the Institute of Moleculaire and Clinical Ophthalmology in Basel, Switzerland. Today we will explore the potential of optogenetics for Usher syndrome patients. Thank you very much for being here, Bence.
Thank you so much for the invitation. It's a pleasure to be here.
Maybe to first understand how the eye works in Usher syndrome patients, we should start off by looking at the healthy situation, because the eye is quite a complex organ, isn't it?
Definitely. So in the healthy situation, light enters through the pupil, and it's focused at the back of the eye, at a thin layer of cells called the retina. At the retina, there are cells that capture this light and convert this to an electrical stimulation. This electrical stimulation is then passed on to other cells, which eventually are passed on to cells that connect to the brain and bring the information from the eye to the brain, where the perception of the image is happening. So as you can see, the retina is not only a camera, but it's also a computer which processes the information that is captured as light.
Yeah. And this is the natural situation. If we then focus on Usher syndrome, what actually causes the vision loss in patients with Usher syndrome?
So Usher syndrome is a genetic disease, and because of mutations in certain genes, certain proteins will be dysfunctional, which will lead to a loss of activity in the photoreceptors which detect light. So essentially, this will lead to a visual impairment, which can progress to blindness in some cases.
Yeah, and is that process to blindness, is that an irreversible process?
So this is a progressive disease, and the visual impairment is gradually getting worse. But it doesn't mean that the cells also degenerate. Actually, we have started a study and looked whether cells in the retina can still be preserved despite having lost their function. And indeed, what we find is that 2/3 of patients, including 2/3 of patients with Usher syndrome, still have inactive cells in the retina, which we call sleeping cells.
So they're not gone, they're still there.
They are still there. They are non-functional. They don't detect light anymore. But we can use ocular imaging technologies to actually capture the presence of these cells in these patients.
Yeah, and that's a good thing, because that is where optogenetics comes in, right?
Exactly. So our research idea is to use optogenetics to resensitize these sleeping cells. Essentially, that means waking them up. That means we would bring in a light-activated protein using a harmless virus to these cells and make them light-activatable again. This protein, what we bring in, is called the optogenetic sensor, or the optogenetic protein. This protein is from algae. It's a simple protein that can detect light. And what we hope to do is to reactivate these dormant sleeping cells by this optogenetic channel, which would allow them to capture light again.
I can imagine that can make a huge difference. What would a patient presumably be able to see again and do again after this treatment?
That's a very good question. This is an artificial vision. This is not natural vision. And the type of vision will depend on which cells you target. There has been a landmark study a few years ago, which was published in Nature Medicine, where researchers targeted ganglion cells, which are the cells that form the optic nerve, which communicate normally between the eye and the brain. These cells normally are not light sensitive, but using optogenetics, you can make them light sensitive. And this is what was done a few years ago. And a patient with Usher syndrome, who has been blind for decades, was treated with this optogenetic vector, and the patient regained some level of vision. The patient could detect objects on a table. The patient could count the number of objects that were placed on the table, and also could touch these objects on a table, despite being blind for decades.
That's a huge difference, I would say. And then your research does not focus on the ganglion cells, the cells of the optic nerve, but on the cells of the retina in the back of the eye. Am I correct?
Yes, so we're going for more upstream. So we're trying to target these sleeping cells that we discussed. And the idea is that if we can target these sleeping cells, we hope we can regain resolution in these patients. That means our aim is not only to restore vision, but also to restore high acuity vision, which allows patients to read.
Read even. That's impressive.
That's the goal. Now we have to say that this will be a monochromatic vision. That means a single color, essentially shades of gray, because we cannot restore color vision. That's an important point. Technology right now is not there. But because we are going for these sleeping cells in the retina that are present in 2/3 of the patients, we hope that in this patient population, we will be able to restore not only vision, but also some level of resolution, allowing patients to read and recognize faces.
Yeah, so it would maybe be an option for many Usher syndrome patients. However, it's not natural vision as we would know it. To what extent do you think we can be optimistic about optogenetics?
I think we can be optimistic. And right now, this is the best hypothesis to restore vision. So there are many technologies that people tried: electric retinal implants, stem cell technology, and optogenetics. These are the three main forms to potentially restore vision. I think because of the simplicity, optogenetics, which uses a single protein, has the highest chance of really being able to restore vision in patients who have lost it. And optogenetics does not depend on the gene. So it can be good for Usher syndrome, but it's far beyond. It's a general way to restore vision.
Yeah, and the way it sounds like is also completely in line with the moonshot goal of Usher Syndrome Foundation. They've set the moonshot goal of making treatment and diagnosis of Usher syndrome standard of care worldwide. How would you say your research specifically contributes to this goal?
So in the case of Usher syndrome, there is a therapy for hearing. There's a restorative therapy for hearing, which are cochlear implants, which really revolutionize the care in this domain. But right now, we are not yet able to restore vision. That's still a goal that has not yet been met. It's an unmet medical need in the world, and it's a highly ambitious goal, which we're trying to tackle. Although I'm very optimistic, we have to be also very careful because this is an experimental therapy. This has never been tested in humans, and we can only say if it's working once we see that the patient recovers vision.
We will follow your research closely, Bence. Thank you for your time.
Thank you so much, and I'm glad to be here.